Hi and Assalamualaikum. For this FIS 3043 Aquatic Botany subject, this lab exercise 5 is about mangrove biodiversity and biological informatics. We are from Group 13 from K1, which stands of Muhammad Hafiz Fikri bin Syed Hashim, Muhammad Ridwan Hakim bin Muhammad Nizam, Nurain binti Mishaw Ghani, and Nur Fatin Fatihah binti Ahmad. So for this lab report, we will explain about the introduction, objectives, materials, method, results, and discussions about the samples about the true mangroves and associates mangrove samples that our group has done.
As plants that only grow in tropical and subtropical, mangrove exist in Malaysia as well, with approximately 575,000 hectares of areas that are identified as mangrove forests. Although named mangrove forests, there are also other species that living besides mangrove species, which are called mangrove-associated plants. These types of plants indicate thriving ecosystem where biodiversity is abundant. They also pose great benefits for both economics and ecological importance. Our first objective is to collect 2 samples from local areas for lab session. Second objective is to examine and identify these samples based on their morphologies. Third is to understand how each species differ with one another.
The materials that we have used is mangrove samples, which is the left side is the true mangrove samples and the right side is the associates mangrove samples. And then for identifying process, our group have used physical books, e-books, and website references for the identification process. The first materials for the identifying process is the hard copy, which is Mangrove Guidebook for Malaysia, which been borrowed from the library. Next for the e-book, which is Flora Bakau Malaysia from UKM. So for the website materials, our group has used a World Mangrove Database for the identification of our mangrove samples. For the method of our group to complete this lab report, I will explain from the samples collection to the samples identification process. So, for the true mangrove and associates mangrove samples, our group have go to 2 different locations, which is for the samples of Soneratia cassiolaris, we have collected it at Kampung Pulau Ketam, Pulau Duyung. Next, for the associates mangrove, which is Barringtonia racemosa, our group have collected the samples from Kampung Bukit Tok Ris. Both are from Kuala Terengganu. Next, each of our group members has their own references on identifying the samples collected, which is from the e-books of Flora Bakau Malaysia and the hard copy books from Mangrove's Guidebook for Malaysia. The hard copy book have a complete data about the samples that we got. Next, the samples have been brought to the lab session for lab report last Wednesday. So at the lab, the group members has identified the sample based on the morphology, fruits, flowers, and leaves samples that shows the specific features of certain species. So from these fruits, flowers, leaves, that samples that we have taken, we identified the species based on the pictures and the key features of the samples. Next, the taxonomy classification and differences between the samples has been discussed by the group members, and we will see at the results.
For this lab, we have 2 mangrove. So first, we look for the true mangrove, which is Soneratia cassiolaris. So for this, we look for the taxonomy classification. For this species, the kingdom is Plantae, subkingdom Viridiplanthae, Infrakingdom Streptophyta, phylum Thraciophyta, subphylum Spermatophytina, class Magnoliopsida, superorder Rosane, order Myrtales, family Lythracia, genus Soniratia, and the last is species Soniratia cassiolaris. Okay, so for this part, we look for the fruit, flower, and leaf. For the fruit, sepals spreading away from the fruits horizontally flat, and it has a big berry fruits melon-like. For the flower, when we look closely, it has a white pom-pom-like, and the thread-like stamen are pink to gradually white. For the leaf, tip of leaf has a mini recurve tip. Okay, so this is the mangrove associate species, which is Barringtonia racemosa. So we look the taxonomy classification: kingdom Plantae, subkingdom Viridae Plantae, infrakingdom Streptophyta, phylum Thraciophyta, subphylum Spermatophytina, class Magnoliopsida, superorder Estrinae, order Ericales, family Lecythidaceae, genus Barringtonia, species Barringtonia racemosa. Okay, now we look for the Barringtonia racemosa for the tree. The tree is a small to medium size. For the fruit, fruit is a water buoyant. It has an egg or oval shape with 4 to 6 fan growth. For the flower, flowers are large, strongly scented, and arranged in spear-like arrangement. Petals are green to pale rose color. For the leaf, leaf edges toothed, leaf cluster at the end of the branches.
As for the comparison between Soneratia alba and our sample, which is Soneratia cassillaris, for Soneratia alba, the filament color is white, but Soneratia cassillaris have green solid color for its filament, and calyx is cup-shaped, enclosing the ripe fruit for S. alba, and calyx is flat, not enclosing the ripe fruit is for Soneratia cassillaris. And for the leaf petiole, Soneratia alba have yellow color for the mature leaf petiole, but Soneratia cassillaris have reddish pink base color. And our next sample, which is Barringtonia racemosa. Barringtonia racemosa has flower with lot of white stamen tipped with either red, white, or yellow. And for the Barringtonia cornelia, which is the sample that the species that we compared to our sample, have white flower is all white except for the pink style. And for the fruit, Barringtonia racemosa have 8 or oval shape and have 4 to 6 fin growth. And Barringtonia cornelia have inverted conical shape and 8 plant protruding edges for its fruit. And for last part is Barringtonia racemosa have spear-like flower arrangement, and Barringtonia cornelia have pom-pom-like flower arrangement.
Okay, now we look for the discussion part. From the result that we found that both of the specimens share most of the attributes with other species under their genus. The identification process become more challenging because of little difference spotted. The only part that can spot the difference are fruit, flower, and the leaf petiole. All the slight difference needed to be observed carefully. For the fruit, the shape and its calyx are the parts that need extra observation because the difference is too little. For the flower, its color is the main thing that can distinguish between its brother from the same genus. The arrangement of the flower also can provide the difference. For the leaf part, the petal can show the difference through its color for our true mangrove species.
So I will explain about the uses of Barringtonia racemosa. It has been used in fishing as a bark and fruits that used as fish poison due to the Barringtonia racemosa contains of saponins. Next, the young leaves and fruits has been eaten as salads or vegetable. Uh, while the Soneratia castellaris has a sour young flesh fruits, which is edible and can be made into pickles. And the fumatophores are also used for corks and making wood soles of shoes. Next, for the Barringtonia racemosa, it grows well when planted inland, and the local name is putat ayam, which is also known as powder puff mangrove associates plants. Next, for the Soneratia castellaris, it has a very strong and vertical pneumatophores, which it always occurs in less saline parts of mangrove forests, which near the banks of the tidal rivers in brackish water. The local name of Soneratia castellaris is known as berembang. So in this discussion, I will focus on Barringtonia racemosa. This tree is native to coastal area of Southeast Asia, Australia, and the Indian subcontinent. Is well known for its very big and dazzling inflorescence. Next, this Barringtonia racemosa has long filaments that extend past the petals of Barringtonia racemosa. The fauna pollination is served as bed food, which they consume the flower nectar. The seeds dispersal method is by the abiotic, which by the water. The trunk of the Barringtonia racemosa tree is a cracked structure of woody type, and the color of the trunk is from grey-brown to pinkish color, while the fruit is in red and green color. So, as I just mentioned just now, the fruit is a fleshy fruit which can be eaten. Next, for Barringtonia racemosa, the tree is able to tolerate for wide range of soil condition. So, this species of tree can live and suitable for most of the sites to grow. And this discussion focused on the Soneratia castellaris samples. It is a samples that come from Litricae family, and this species of tree has a whipping branches that grow up to 15 meters tall with powerful vertical pneumatophore. And this tree with light to dark brown bark have a fractured texture on their older trunks, where the younger trees have tentacles bark. A well-developed glossy fruit with a reddish pink base and a well-developed pneumatophores with a red and white stamen that are red in lower and white in higher portion for the flowers. The sepals are large and it is difficult to grip to the fruit because the sepals are flatly and horizontal expanding out of the fruit. So this species of roots resembles spear-shaped pneumatophores in that they are thin and pointed tree.
Our first problem is due to the height of the trees of Barringtonia racemosa and Soneratia castellaris. No flowers can be collected. Second problem is we are unable to collect leaf samples for Barringtonia racemosa as vines that help fruits lack leaves. Lastly, we require permission from locals to collect samples as both specimens grew near local jetty.
conclusion, the morphological characteristics of true mangrove plant and mangrove associated plant were able to be examined using its flowers, fruits, and leaves. This play a vital role in order to identify the species for each sample obtained. Besides, the identification of the species obtained was done successfully using all of the morphological characteristics found. In addition, the differences between the species that shares the most identical attribute were able to be observed. It is due to the it is due to its morphological characteristic that differ slightly between the sample that have been observed and other species from the same genus that made it possible.
So this is the references that we have used for our lab report, which is book and ebook for identifying process and online articles for our more informations. Same goes to the website references, which is the first and second websites are used for the identifying process, and next website are used for the description about the samples.